The overall goal of this protocol is to investigate the cellular interactions between human corneal stromal cells and neuronal cells in their natural extracellular matrix. The protocol can be expanded to study corneal diseases and dystrophies such as keratoconus, diabetic keratopathy, and Fuchs dystrophy.
This method can help answer key questions in the human cornea biology field by identifying the underlying mechanisms and defects in the stromal-nerve interactions during a specific cornea disease or dystrophy. The main advantage of this technique is that cells are allowed to secrete their own extracellular matrix in vitro that provides a better and more accurate model of the actual human cornea tissue in vivo.
The implications of this technique extend toward therapy of corneal diseases because assessment of cell-cell and cell extracellular matrix interactions will ensure accurate representation of the corneal tissue and allow a more targeted drug development.
Though this method can provide insight into corneal nerve damage and regeneration, it can also be applied to other systems, such as drug screening and development.
Upon receipt of human cornea tissue samples, transfer the tissues into a 21.5 square centimeter dish containing 2 milliliters of sterile Dolbeco's phosphate buffered saline. Position the cornea dome face up and use a razor blade at a 45-degree angle to scrape off the epithelial layer. Next, flip the cornea dome face down and thoroughly scrape off the endothelium from the stroma. Wash the stromal tissue with DPBS. Keep the stromal pieces wet with DPBS and use a number 10 surgical